Thanks. So I'm going to start by saying that my talk is non-apologetically phenomic. I have very little genome data at the moment, but I want to emphasize that for the questions that I have been, wow, that's really loud, okay. For the questions I've been going after, I think actually taking this more phenotype approach has been necessary. And I'm hoping, after listening to all of these great talks here today and drooling over all of these nice metagenomic virome analyses, that I can start bridging the two and marrying them. So I'm going to be talking about bacteriophage interactions in the plant phylosphere. I really like the plant phylosphere as a model system, but I actually do treat it as a model system. I'm trying to fundamentally understand how phages interact with microbiomes, and plants are particularly nice because they stay put and you can sample them over time, and there's, I would say, far less ethical considerations. You can treat them in any way you see fit, and they don't tend to tell you they don't like it. I'm a disease ecologist, so my interest in phage is very much coming from a disease ecology perspective, trying to understand what makes hosts susceptible to pathogens. And I'm interested both in why bacteria are susceptible to bacteriophages, but moreover, why eukaryotic organisms are susceptible to bacteria and what role phages might play in that. And so I feel like this figure here summarizes a lot of work that is quite recent in the field. So it's been long known that hosts genetically vary and that this influences their susceptibility to disease, but now it's clear that that also influences what microbes we're colonized by in terms of our microbiome. And we have long known that pathogens co-evolve with host populations, but now it's seeming like pathogens also co-evolve with the microbiome, with our commensal bacteria, and that's very exciting. And then finally, the recent last 10 years or so, it's become clear that the microbiome is actually a critical part of our immunity, our ability to defend ourselves against pathogens. And this is, to me, very exciting and completely changing agriculture and medicine. But only in the last sort of 2 years, I would say, have people been extending this to all the phages that potentially are also influencing the microbiome and how that might impact disease susceptibility of the hosts. So that's the broad umbrella under which most of the work I'm doing is going to fall. And I'm going to talk about, first, a natural system that I spent most of my postdoc working on and have a couple of unpublished results at the end. And then I'll tell you quickly about the model system we're developing in my lab now. So I wanted to work on a system, as I said, that stood still and I could sample over time, and I decided to work on trees because they're also long-lived organisms like us, and I thought that would be a nice place to look at how the microbiome might confer resistance to pathogens, given the high discrepancy in timescales and generation times between trees and pathogens. And the system that I had been working on is horse chestnut bleeding canker disease, and as I said, all phenotype for this first part of the talk. I was looking at the culturable bacteria that I could get out of leaves and any plaque-forming phages that came out of those same trees, those same leaves. And to summarize a whole lot of work, the first key result that I found is that the phylosphere of trees is chock full of bacteriophages. No surprise there. And these phages are very well adapted to their bacterial hosts. What I mean by that is if I take multiple leaves from the same tree, I culture the bacteria from them and then culture any phages from them by filtration and do cross-inoculations, traditional microbiology soft agar overlays, and just simply ask who's susceptible to whom, it turns out that within a tree, phages are equally infective to bacteria from one leaf or another, but if you compare phages from one tree to phages from a neighboring tree of the same species, another horse chestnut tree, phages are highly locally adapted. They're much more infective to bacteria from that same tree than a neighboring tree. And this isn't surprising, right? Because different hosts have very different microbiomes. I've already said that. And it's true that, if you look just at the top here across my 8 different trees that I sampled for this experiment, they did have different phylosphere communities. And importantly, those different phylosphere bacteria differed in their susceptibility to phage within that same environment. But what's also quite nice, and I don't show the data here, is that these bacteriophages were even well adapted to a single bacterial species. So if I just look at Pseudomonas syringae, the pathogen, phages were much more effective to their local Pseudomonas syringae than to Pseudomonas syringae from a neighboring host. So I was quite excited about this because it says that phages are there and potentially important. And so I went after the next key question, which is, does that mean anything for the bacteria? Are they then selecting for bacterial resistance over time? And because we can take these, bring them into the lab and freeze them, we were able to do these time shift experiments. And by that I mean that I can take bacteria that I pulled out of the leaf in July, and I can ask it how susceptible it is to phage from the same time point, July, earlier time points, June, May, and later time points, August, September. And I collapse all that data, and why I'm doing all this is simply to look to see whether there's a linear relationship, or a relationship at all, between the time shift and bacterial resistance. So if bacteria are evolving resistance to phage, as you'd expect based on their level of adaptation, you would expect that bacteria are more resistant to phages from the past, the selection pressure which they have experienced, and less resistant to phages from the future, those that have counter-adapted to any resistance that's around. And from these natural populations, just like we see in the lab, that's exactly what we found. So on average, bacterial resistance is highest against phages from the past and lowest against phage from the future. And I think this is actually a non-trivial result. I mean, it's intuitive and it makes sense and it's exactly what I predicted, but bacteria in nature are experiencing an onslaught of selection pressures, not like our nice little microcosm lab experiments. And so I think it's really critical to show that these lytic phages in this environment are important, even in light of all of the other selection pressure that bacteria are experiencing. And I put this up here just to say that host range and who's infecting who is a complex issue, and Matt alluded to this earlier in his beautiful talk, but these are just a couple, this is a very small preliminary experiment we did, where we took some of our phages from the horse chestnut phylosphere, tested them against different bacterial isolates that we had in the lab that were either Pseudomonas syringae or also on the bottom there, Erwinia. And what we found is that it looks quite modular, for those of you who follow these bipartite networks, but also that some of these phages, a single phage type that we pull out, can infect both Pseudomonas and Erwinia. But interestingly, it doesn't, just because they have a broad host range and can infect both Pseudomonas and Erwinia, doesn't mean they infect all our Pseudomonad syringae isolates. So host range is complex, and I would say that it's also complex. Whoops. Oh, I must not have put that in here. Sorry, I'll come back to that. Anyhow, it's also complex, and maybe I'll come back to this, so maybe my slides went out of order, but building on what Matt showed, if we take the same phage and test it on multiple bacterial hosts from the phylosphere, we get very different PFU counts. And so we've also discovered that same result where, yeah, bacteria are not either susceptible or resistant again. So the host range is a very complex measure, and we're not very good at estimating it, I would say. So the last bit of data that I worked on in this system was combining these sort of local adaptation spatial analyses with time shift temporal analyses to do large crosses between bacteria and phage that were isolated from either the same or different trees and the same or different time points. So it was a huge amount of work, but it basically can be summarized just in one graph. If we look at phages from September, from the end of the season, and again, these are lytic bacteriophages, or at least when I caught them, they were lytic. Phages from September, and we ask who they can infect in terms of bacteria that are either sympatric from the same tree or allopatric from another tree, and then from earlier in time. What we find is that, again, we recover this pattern where they're more infective to their local bacteria, as I saw previously. But what's interesting here is that phages are most infective to bacteria that from a month earlier, but their infectivity wanes, if you will. So it's not that they've just become overall more infective, that it was a sort of an arms race dynamic. They've lost the ability to infect bacteria from much earlier in the season. And this is important. This is, as a disease ecologist, this is very exciting. It's what we call fluctuating selection dynamics. This drives diversity. It's one of those mechanisms that we get really excited about as evolutionary biologists and ecologists. But also it tells us something nice about how bacteriophage are interacting. And it means that, that you wouldn't necessarily expect a phage to stick around forever. At some point, it's losing its ability to infect bacteria that were around previously, so those bacteria can reinvade that population. So we did the same experiment, but we flipped it on its head. And this was work done by Nicole Parr, who was a research technician with me at the time. And we took, in this case, we just did the opposite experiment. We took bacteria from September, and we challenged them against phages from either the same or different hosts and from past time points to see, again, whether resistance has that sort of short-lived nature, that fluctuating selection. And we saw exactly the opposite. So here, bacterial resistance does change. Bacteria are slightly more resistant to phages that they've experienced in the past, as I showed you previously. But now it doesn't wane. They're still resistant to phages from the beginning of the season. And we thought, okay, well, maybe that's just a generation time thing, right? Maybe it just is that the time scales are different. Phages evolve very quickly, so they lose those mutations more readily than the bacteria. So we actually did this over 4 years now. And if you just look on the bottom here, I've left those two graphs that were within a season on the top. If you look at the figures on the bottom, we see generally the same pattern. So if you take phages from 2014, they lose their ability to infect bacteria from very far back in time. The error bars are huge, and I'm happy to talk about why that's the case. But of course, because we saw so much variation within a season, we would expect such large variation if we're going across years. But for the bacteria, we still see this long, durable bacterial resistance. I don't entirely know what it means, and if anyone in the audience who thinks about bacterial resistance mechanisms, Matt, if you want to chat afterwards about what this means, that would be terrific, because I'm excited about this result, but I haven't quite resolved how you can have both of these patterns happening simultaneously. So the take-home message from that horse chestnut system is that using these sort of phenotypic approaches, where we look at bacteriophage infection of bacteria in terms of the culturable subsection, what we can see is that phages are well adapted and they select for resistance. So they're important in these realistic environments. And phage infectivity seems more specific than bacterial resistance. And again, this might be expected under some CRISPR-Cas scenarios, but I haven't fully explored this pattern. So we've been moving into a model system to try to make it a little bit less black boxy. And we started with a very, very simple experiment. This is done by my PhD student at the time, Sean Meaden. And he just took these spontaneously resistant mutants to 6 different phages. And so this googly eye thing is supposed to represent a petri dish where we're putting on high titers of phage, and then every now and again in that presence of high phage titer, you'll see a colony pop up. We then confirm that it's resistant. It should be a single mutational step. And then we used those mutants to ask questions about how resistance evolves, and in this case, specifically about how costly that resistance will be to the bacterium. And so just to show proof of concept here, yes, the resistant mutants, if you put them in liquid media with the phage, they grow better than the ancestor who's not resistant. No surprise there. Resistance was selected for. If you now grow them in the absence of phage, what we observed was that there was no cost of resistance if you grow them in a cushy lab environment. This is not under competition. These are grown as monocultures. They grow equally well as the ancestor. There we go. In a test tube, but when you put them back into this naturally complex and highly competitive plant environment, in this case a tomato, you recover that you see a cost of resistance. So these phage-resistant mutants are not able to grow as well in the plant phylosphere. And this leads to a prediction, which is, and really this motivated the entire work, is what we are seeing in a test tube where we see bacterial resistance evolving very rapidly and via very simple mutations, as Matt previously alluded to. Is that predictive in any way of what's happening in a more natural environment? And so my PhD student, Cathy Hernandez, since it's a genome, here's a genome. We genome resequenced everything, and sure enough, we could uncover that most of the mutations we were observing were mutations involved in the LPS pathway, and so there's clear reason to predict this cost of resistance that we observed. So Cathy Hernandez has been following up on this question by taking the same bacterium, same phage, but passaging them in tomato for very long periods of time. And she has done this experiment numerous times and under many different MOIs and settings. And what we are continuously observing, and sorry, the text here is a bit small. What you're looking at here is in a test tube. Here is when you don't evolve them with phage. This is susceptible bacteria, and these two colors are different types of resistance. In the absence of phage, no resistance evolves. But in a test tube, if you put phage in there and you either evolve them or allow them to co-evolve, resistance rapidly spreads. But in a plant, same phage, same MOIs, this is just one experiment out of many, we just do not see resistance spreading at all. And so, you could argue this is a clear juxtaposition to what I just told you for what's happening in the natural phylosphere. But what this basically tells you is, at least in this case, and we're now trying to figure out how widespread this is, that cost of resistance is actually very important. It suggests that resistance can evolve very rapidly, but that might not be selected for in a more complex plant environment. And from a phage therapy perspective, going back to Vivek's talk, this is quite important and exciting, as it means that we don't maybe have to worry as much as we do about bacterial evolving resistance to phages. And then, how much time do I have, Maureen, for this last story? Perfect. Oh, okay. Well, in that case, I'll go. So the last work here is actually work that's done by Norma Marella, and she's presenting a poster. So I'm going to leave out some really fun and critical details of this part of the talk that you should go and see her poster for. In fact, I put a big white box over one of the key results just to make sure that you do. But what we've been doing, again, to bring this back into the lab, but to add a little bit of complexity, is going to natural microbiomes. And by this, I mean that what we do is we go to tomato fields at UC Davis. We collect samples of tomato grown in a natural field setting, and we sonicate off all of the phylosphere microbiota. So all the bacteria, fungi, archaea, unicellular eukaryotes, whatever's coming off the phylosphere, the above ground portion of these plants, and of course the viruses. And then through a series of filtration steps, Norma was able to separate this microbiome into the bacterial and fungal component, the viral fraction, and then the subviral fraction. And we had a very simple question we wanted to address, which is, we know the phylosphere is full of phages. Does this actually matter in terms of colonization ability? So we're very interested in my lab at the moment about microbiome transmission, and so we're curious as to whether phages play a role in initial microbiome establishment. So she did this filtration, and then she re-inoculated now mostly sterile tomato plants, juvenile plants, with either just the bacterial fungal component or with that component, the microbiome, combined with the viral fraction. We sprayed them onto the new plants, and then we simply ask what happens in terms of both bacterial abundance and bacterial diversity. So in terms of abundance, what you see here is that when you co-inoculate with the natural phage fraction, bacterial abundance after 24 hours in the phylosphere is much lower than in the absence of phage. So again, it's not surprising, but it's nice to confirm that these phages, these lytic phages, really matter. They matter in terms of whether bacteria can grow rapidly when newly colonizing a host. No, I won't give away. There's also a day 7 result that you'll have to go to the poster for. And now here, what you're looking at is 16S community profiling in the presence and absence of phage. And again, after day 1, in terms of looking at who's colonizing, what this microbiome looks like during that early stage of succession, whether or not phages are there really matters. And what does this mean in terms of microbiome diversity? And we care a lot about microbiome diversity because we know that's highly correlated with disease status and dysbiosis, if you believe in such a thing. And so what we're looking at here is the alpha diversity. Whoops, sorry about that. Still trying to figure this. Yeah, alpha diversity on the top, so how much diversity there is within a plant, and then beta diversity on the bottom, how different plants are plant replicates are within a treatment. And what we see is that phages increase alpha diversity, and this is as you would expect based on kill the winner model, for example, that phages increase diversity by targeting common bacteria and decreasing their fitness, thereby allowing more rare species to increase in density, but actually decreases among host diversity. So phages seem to be, when phages do select, they actually make microbiomes more similar to one another than when the phages aren't there, which I think is quite exciting. Oh, and then there's a third treatment here, which is the subviral fraction, and I will point you all to Norma's poster to figure out the cool thing that happened there. All right. So just to wrap things up here, as I told you, my motivation for all of this work here is to really understand the role that the phages play in the microbiome. There's this huge interest in microbiome at the moment, and what we know is the microbiome is complex within hosts. It's variable across space, such that each of us have different microbiota. It's dynamic across time, and it's important in shaping host phenotype. But I guess what I'm emphasizing in this talk with the data, but also is becoming more clear even in the human virome literature, is that phages are known to play a role in all of these processes. So we just can't ignore them. So I've shown you that phages can shape the composition of bacterial communities. They can show specific adaptations to bacteria, therefore maybe driving two hosts apart. They can drive changes in community composition over time. And of course, phages are fundamentally important in shaping bacterial phenotype. If we're saying that bacteria are important to our phenotype, well, you do the math. And with that, I will take questions.